nanometers due to absorption of an aromatic amino acid. Tryptophan phenylalanine tyrosine is now well established that killing of cells by UV radiation is due primarily to its action on DNA, and so UV radiation at 260 nanometers is most effective as a lethal agent, although several effects are known. On well-established effects is in the induction of DNA of pyrimidine dimers at state in which two adjacent pyrimidine bases, cytosine and thymine, become covalently joined so that during replication of the DNA, the probability of DNA polymerase inserting an incorrect nucleotide at this position is greatly increased. The type of UV radiation source most frequently used for mutagenesis is the germicidal lamp, which emits large amounts of UV radiation in a 260 nanometer region. A dose of UV radiation is used that brings about 90–95% killing of the cell population. And mutants are then looked for among the survivors. If much higher doses of radiation are used, the number of visible viable cells will be too low, whereas if lower doses are used, insufficient damage to the DNA will be induced. UV radiation is a very useful tool in isolating mutants of microbial cultures. Ionizing radiation. Ionizing radiation is a more powerful form of radiation and includes short wavelength rays such as X-rays and cosmic rays and gamma rays. These radiations cause water and other substances to ionize. The mutagenic effects are brought about indirectly through this ionization. Among the potent chemical species formed by ionizing radiation are chemical free radicals, of which the most important is the hydroxyl radical, and inactivate macromolecules in the cell, of which the most important is DNA. DNA is probably more sensitive to ionizing radiation than other macromolecules, but because DNA is the genetic material, change is induced and it can have a permanent effect. At low doses of ionizing radiation, only a few hits on DNA occur, but at higher doses, multiple hits occur, leading to the death of the cell. In contrast to UV radiation, ionizing radiation penetrates readily through glass and other materials. Because of this, ionizing radiation is used frequently to induce mutations in animals and plants, whereas its penetrating power makes it possible to reach the gamete-producing cells of these organisms readily. But because ionizing radiation is more dangerous to use and is less readily available, it finds less use with microorganisms where penetration with UV is not a problem. Mutations that arise from DNA. Recall that mutation is an inheritable change in the genetic material. Therefore, if an error in DNA synthesis can be corrected before the cell divides, there will be no mutation. Furthermore, some DNA damage clearly cannot be replicated and therefore cannot itself be mutation. For instance, if a DNA molecule contains pyrimidine dimers, it will not be replicated to give two DNA molecules, each containing pyrimidine dimers. If such DNA damage cannot be repaired, cells often die. Most cells have a variety of different DNA repair processes to correct mistakes or repair damage. Many of these DNA repair systems do not make mistakes. However, some processes seem to be error-prone, and it is in the repair process itself that introduces the mutation. Many kinds of mutations arise as a result of faulty repair of damage induced in their DNA by some of the various agents just discussed. Often DNA damage itself can induce DNA repair system, and a complex cellular mechanism called the SOS regulatory system is activated as a result of some types of DNA damage, initiating a number of DNA repair processes. However, in an SOS system, some DNA repair occurs in the absence of a template instruction, that is, without base pairing, which results in the creation of many errors, hence many mutations. In the SOS regulatory system, DNA damage serves as a distress signal to the cell, resulting in the coordinate depression. Derepression, induction of the number of cellular functions involved in DNA repair. The SOS system is normally repressed by a protein called LEX-A. However, this repressor protein is inactivated by REC-A, a protease that is activated as a result of DNA damage. Since one of the DNA repair mechanisms of the SOS system is inherently error-prone, many mutations arise. Thus, though the SOS regulatory system DNA damage by various agents such as chemicals and radiation leads to mutagenesis. Much of this is brought about by DNA polymerase B, which is encoded by the UMC and UMUD genes. The error-prone DNA polymerase IV is also induced as part of the SOS response. These enzymes are sometimes referred to as DNA mutases. Once the DNA damage has been repaired, the SOS system is switched off and the further mutagenesis ceases. In addition to its effects on the cellular mutation rate, the SOS regulatory system plays a central role in the regulation of temperate virus replication. It should be emphasized that not all DNA repair occurs in the absence of template instruction. Cells generally have many DNA repair systems that require template instruction and lead to proper DNA repair. These systems apparently work most of the time, but are not sufficient to repair the large amounts of damage done by some of the agents previously mentioned. Changes in the mutation rate. The mutation rate of an organism is a phenotype under selection pressure like any other. The fact that the hyperthermophilic archaea have about the same mutation rate as laboratory strains of Escherichia coli might lead one to imagine that this pressure had led to the selection of organisms with the lowest possible mutation rates. However, this is not so. Mutants of some organisms are known that are hyper-accurate, and these organisms, though the proofreading or repair mechanism may have seen a high metabolic cost, that the organism is typically at a disadvantage. Of course, certain organisms may benefit from such mechanisms to accurately certain environmental niches. A good example is bacterium Deinococcus radiodurans, which is 20 times more resistant to the ultraviolet light and 200 times more resistant to ionizing radiation than E. coli. The resistance depended in part upon redundant DNA repair systems and on the mechanism. For example, exporting damaged nucleotides allows the organism to survive in environments where other organisms cannot. Perhaps even more interesting, the opposite seems also be true. That is, some organisms may benefit from an increased mutation rate. DNA repair systems are themselves encoded by genes that are thus subject to mutation. For example, the subunit of DNA polymerase III involved in proofreading is encoded by the gene DNA Q. Certain mutations in the gene lead to cells that are still viable but have an increased rate of mutation. Strain with an increased mutation rate is said to have hyperpermutable or mutator. Mutations leading to a mutator phenotype are known as a number of DNA repair systems. The mutator phenotype is apparently selected for in complex of changing environments, since strains of bacteria with mutator phenotypes are more abundant under these conditions. Presumably, such environments, whatever disadvantage increased mutations rate, may have its offset by the ability to generate new and useful mutations. Note that when the environmental conditions stabilize or return to normal, the advantages of the mutator strains will be lost. As we have indicated earlier, a mutator phenotype may be induced in a wild-type strains by stress situations not because of mutations in the genes involved in the DNA repair, but because of the normal regulation of function of these genes. For instance, the SOS response induces error-prone repair. Therefore, when the SOS response is activated, the mutation rate increases, while in some cases this may be a necessary byproduct of DNA repair. In other cases, the increased mutation rate may itself be of value. This is, the organism may generate adaptive mutations. Adaptive mutations lead to a phenotype and a mutant that allows it to survive a particular stress. Biological and site-directed mutagenesis. Mutations can be introduced without the use of chemical or physical agents through the process of transposon mutagenesis. We discuss this detail of transposon mutagenesis later in this chapter, and note here only that if insertion of transposable elements occurs within a gene, loss of gene function generally results. Because transposable elements can enter the chromosome at various locations, transposons are widely used by microbiology genetics as mutagenic agents. So far, the mutations that we have been discussing have been randomly directed at the genome of the microbial cell. Recombinant DNA technology and the use of synthetic DNA make it possible to introduce specific mutations in specific genes. The procedures for carrying out the mutagenesis of specific sites in the genomes are called site-directed mutagenesis and will be discussed in detail at the end of this chapter. Mutagenesis and carcinogenesis, the Ames test. A practical use of mutant bacteria strains that has been developed to identify potentially hazardous chemicals in the environment. Because the sensitivity with which selectable mutants can be detected in large populations of bacteria is very high, bacteria can be used as screening agents for potential mutagenicity of chemicals. This is relevant because it has been found that many mutagenic chemicals are also carcinogenic, capable of causing cancer in animals or humans. The variety of chemicals, both nature and artificial, that the human population comes into contact with through agriculture and industrial exposure is enormous. Therefore, a considerable need for a simple test to ascertain the safety of such compounds. There is good evidence that a large proportion of human cancers have environmental causes, most likely through the agency of various chemicals, making the detection of chemical carcinogens urgent. It does not necessarily follow that because a compound is mutagenic, it is also carcinogenic. The correction, however, is very high, and the knowledge that compound mutagenic in bacteria system serves as a warning of possible danger. Similarly, the fact that a compound is not mutagenic in bacteria system does not mean that it is not carcinogenic, because the bacterial system cannot detect all compounds active in higher animals. The development of bacterial tests for carcinogenic screening was carried out primarily